Hello, Dr. Alaka, Mura, and Olatini. We'll be starting in 10 minutes. Thank you for joining. And meanwhile, we begin. I would like to hear from you about your research interests and also your background. You can put it in the chat box. Looking forward to hear from you. Hello, everyone. I would like to welcome those who have just joined. We'll be waiting for everyone to join and then we'll be starting in 5 more minutes. Thank you for joining. And meanwhile, we begin with the webinar. It would be great to hear from you about your research interests and your educational background as well. Please feel free to put in the chat box about your interests and your background. Also, if you have any questions, you can please put them in the chat box. Hello, everyone. I would like to welcome those who have just joined. We'll be starting in more 2 minutes. We are waiting for others to join as well. A very warm good morning, good afternoon, or good evening in whichever part of the world you are. So let's begin with today's webinar. I would first of all like to welcome you all to this webinar. We'll be talking about the summer training programs that will be starting from next week. So today we are going to talk about introduction to bioinformatics, Omixlogic transcriptomics, Cheminformatics and structural biology, and also the Industry Academia Fellowship with Omixlogic under mentor guidance. So before we start talking about each one of these programs, I would just like to quickly introduce us for the ones who have joined us for the very first time. So we are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools. And our mission is to make bioinformatics more accessible. The training has been completed by over 24,000 participants from 187 countries in over 350 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The Omixlogic training follows a project-based learning approach, which uses research-grade tools to analyze data from top peer-reviewed journals. Omixlogic training has been completed by 6 different specialization tracks in oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Now, I would like to introduce our team founders and co-founders as well. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of information. And one of his major projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people who are mentioned here on the right. Julia Panov is an expert bioinformatician. Avi Titavsky leads the development of the T-Bioinfo platform. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals, and he has been developing collaborations with universities as a mentor guide and project lead for Omixlogic training and research fellowship. Dr. Raghavendran Lakshminarayanan is an accomplished computational biologist with an experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. I'm Sonalika Ray and I'm the Omiclogic community manager, and I'm responsible for daily interaction with thousands of users that are a part of our team, Buying Biotech. To introduce Farshthar, she's our sales executive and she'll be helping you enroll in any of the programs that you are interested in. And Ojasvi helps us with the web analytics. So today, let's talk about bioinformatics first, and we'll also discuss about the programs, the structure, and the resources that you can leverage the most. So what is bioinformatics? It is the development, implementation, and usage of methods and software tools for understanding biological data. Bioinformatics has practical applications in healthcare, agriculture, and mitigating climate change. As this field progresses, bioinformatics poses several challenges for both students trying to learn it and faculty trying to teach it. So in this presentation, I will explain the current challenges and limitation of bioinformatics. I would describe how we aim to address these challenges with the Omixlogic Learn portal. I would describe the Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework. We'll explore the Omixlogic portal and how it utilizes the Bloom's taxonomy in structuring its courses, pipelines, and projects, and finally describe several outcomes of the Omixlogic portal. So what are some of the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The data sets that are analyzed are usually very large and take immense amounts of computing power and memory that many personal computers do not have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs required to do so. In a university setting, students have the opportunity to learn the background information of designing and implementing a bioinformatics project, but do not receive practical hands-on experience with full workflow due to the limitations mentioned previously. Similarly, faculty faces the challenges Similarly, faculty faces the challenges with being able to cover a diverse range of topics if they are specialized in a certain area of bioinformatics or if they're unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know about the metagenomics of seawater. And to overcome these challenges, the Omixlogic Portal was developed. This program aims to standardize, organize, and systematize bioinformatics training in order to make these analyses usable for any biologist or clinician. There are some traditional problems with the standardization of bioinformatics coursework. Materials for courses are usually developed in-house, taught sporadically, and are rarely updated. The Omixlogic portal introduces new material that keeps up with new technology. These courses also need to be organized because the challenge is to combine several different topics together. The flow and transition between subjects needs to be developed, and this is addressed at Omixlogic through introductory material being woven throughout course. The last challenge we face is systematization. How can you know that someone is stuck and needs help or allow someone to run forward? To address this, Omixlogic uses standardized modules and interactive sessions beginning with mastering terminology and concepts to implementing these concepts with real data sets in a project-based approach. Here is an overview of the learning process. In order to create a clear path of skill development, Omixlogic employs the Bloom's taxonomy levels of learning. This approach starts with the basics. Students recall facts and terminology and are able to repeat and memorize the material. With the base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The students will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do Omixlogic resources help advance bioinformatics? Omixlogic is a portal for practical and theoretical learning of bioinformatics, composed of courses designed to take you through the theoretical knowledge. Omixlogic takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give a student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, Omixlogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or beginning a data analysis project altogether. These 6 courses that we'll be talking about in this slide were taken from the core Omixlogic curriculum, which is available online. Beginning with an introduction to the applications of bioinformatics and an overview of data types, the student is able to learn terminology and factual knowledge to build upon. From there, genomics, transcriptomics, metagenomics, epigenomics, and data science are introduced. Each course starts with important terms and concepts and then introduces case studies and pipelines related to each other. And this allows the students to gain conceptual knowledge in order to begin their own projects. Omixlogic expands upon its basic curriculum with specialization track projects. Bioinformatics methods are best learned in the context of real case studies, which are prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research in medicine, biotechnology, agriculture, and even astrobiology. And the projects are prepared by taking publicly available datasets and using them to explore and understand both the research challenge and also the analysis path to extract meaningful insights from complex omics data. The portal gives students the options for analyzing a given dataset, code-dependent paths or code-independent paths. These datasets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly T-Bioinfo platform for big data analysis. In a while, we'll be taking you through the portal as well. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in popular languages like R and Python in order to create your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs, where experts share their experience and support research projects which are developed by you. Now, here is an example of the code-independent pipeline. The pipeline builder interface on the T-Bioinfo platform is a color-coded pipeline that helps establish a logical connection between the technical names of data analysis methods and the logical steps that are needed to build the full pipeline. Data is put in as an input file, and then analysis can be chosen based off of the type of research. Each analysis is separated into 3 colors: blue for pre-processing, red for mapping, and green for quantification. Each time you click a button, a pop-up will contain additional information which is useful for building the pipeline. The ease of color-coded buttons allow programs to be run without any coding knowledge. Interactive outputs allow each visualization of the data. Several demo pipelines with real datasets are available for students to practice the workflow, including demos on genomic variant calling for cancer cell lines. 16 is amplicon sequencing, an analysis on diet and its effect on the microbiome, and a pipeline for examining a phylogenetic analysis of Ebola virus. Shortly, we'll be taking you through these demo pipelines as well. Now, after using the T-Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge, the remember and understanding levels of Bloom's, and apply that in a project. They apply, analyze, and evaluate levels of Bloom's. Then check their comprehension with levels of with different levels and badges, certificates of completion, and test. Similarly, faculty should be able to assign the Omixlogic portal to their students and also design a holistic and comprehensive knowledge of bioinformatics, beginning with bytes and molecules and on to designing their own question-based project. Omixlogic employs a hybrid approach, combining learning and research, and to ensure that the students are building off of the first level of Bloom's taxonomy. Omixlogic tracks progress with activities like pipelines, coding, and literature reviews, quizzes on topics, key terms, and important questions, and covers a broad range of topics like biomedical research, biotechnology, and agriculture. Then students are able to continue their knowledge base with the research assessment, making sure they are able to understand methods, challenges, and results, as well as being able to comprehend primary research literature and recreate the methodology. They should be able to ask questions based on data exploration, and this leads to the ability to encounter errors in their analysis, apply theoretical knowledge of why those errors are occurring, and find a solution. Bioinformatics combines life science terms with data science skills, which allows researchers to answer life science questions with reproducible data analysis methods. The portal, we are proud to collaborate with many diverse programs across the globe. We also work with over 24,000 students at community colleges like Glendale Community College, top-tier universities like Georgetown University, as well as 20 universities around the world. And if you want to learn more about it, then you can definitely get started on the Omiclogic Learn portal, which is absolutely free. and anyone can register for free with many courses available. And now let's begin by talking about the different summer programs that we wanted to discuss in this free webinar. So to begin with, I would now like to pass on the stage to Mr. Ilya Brodsky. Mr. Ilya Brodsky, CEO Pine Biotech, could not make up for the meeting, and he has shared with us a recording. And I would like to invite Mr. Ujasvi Datta to please play that recording for us. Ujasvi, can you share your screen?
Yes.
Kruti, sure, we can share the Slack link. Just give us a couple of minutes, we'll be sharing. Over to you, Ujasvi.
So, can you see my screen? Is it visible to all?
Yes, you can see your screen.
Bioinformatics is the intersection of informatics, or the science of processing data for storage and retrieval, and biology, the study of living organisms. Why should you care about bioinformatics? There are a few reasons for that, even if you're not in computer science or IT. More and more data is being generated in the research and biomedical fields. This includes agriculture, biotechnology, and biomedical research. So knowing what kinds of data are being generated and how it is being analyzed will bring value to your personal career and the ability to collaborate effectively with others. So let's briefly talk about what is bioinformatics. Bioinformatics is a conversion of biology and data, where both are equally important, as we will see in the next few slides. Who uses bioinformatics and who is this for? The answer might have been statisticians, computer scientists, or engineers. But the truth is that more and more biologists and clinicians are now expected to understand and handle their own data, and to publish and succeed in basic and translational research as well as industry. In fact, you can say today that biology itself is rapidly acquiring the characteristics of a data science. This field developed tremendously in the last several decades, basically transforming life sciences, enabling technology, and empowering biomedical research at scale. As a result, many new discoveries that include new treatments, diagnostic assays, design strategies for experimentation were discovered and perfected. As a result, there is a growing interest among biologists and clinicians in data, including new types of data generation, like different types of omics technologies, new methods of computational analysis, as well as approaches that allow us to mine complex data to find patterns that can explain biological phenomena. A lot of breakthroughs in bioinformatics are a result of the growth and adoption of high-throughput technologies. These technologies generate billions of data points on genes, proteins, and other many molecules that can characterize biological systems, how they function, and what are the different variations from normal or different types of clinical conditions that we can describe using these techniques. The data are complicated, but they're organized and compiled into repositories that can be systematically studied to understand, interrogate, and establish trends for many different applications. One such repository is maintained by the National Center for Biotechnology Information, which has a collection of peer-reviewed articles, biological datasets, and projects on a variety of topics. Introduction to Bioinformatics is a program that is designed to introduce you to these important terms and topics. So after completing the course, you will be able to see the applications of next-generation sequencing, biostatistical methods of analysis, and other computational tools that are used to process and mine big data, such as machine learning. More importantly, the program is more than just a theoretical introduction. It's really a practical course. It will give you a sense of what types of technologies are used, and we call them omics technologies, what kinds of analytical methods are used by people in the industry and in research to analyze and interrogate these data sets. And most importantly, you will have access to a portal and a community that will support your journey. So the training program is going to be run through this Omixlogic portal. And the Omixlogic portal combines many important elements that you need to succeed as a beginner in bioinformatics. It gives students options for analyzing a dataset based on code-dependent paths or code-independent paths. So you can pursue bioinformatics by learning coding languages like R and Python, or you can choose to just understand the logic of analysis by not necessarily dedicating time to that. The portal offers many different ways to study biology using data at different levels, complexity. So what you will find is a way to get started, then identify a type of data, a type of omics data like genomics, epigenomics, or others, and then pursue potentially a more technical skill enrichment path that might include coding and statistical analysis. But you can also skip those and especially focus on projects, which are publications designed to introduce you to a real scientific challenge that you can study using data. You will find collections of training modules that are organized under this Getting Started collection. So the program is designed to point you to the relevant resources and complete them over the course of the program that we are going to start. The resources will introduce bioinformatics applications in healthcare, biomedical research, biotechnology, and agriculture, provide you with a concise overview of various biological concepts that you need to know to understand omics data, introduce you to big data analysis using several example datasets, and expose you to some coding in R and Python so that you are aware of how the analysis of biological data is done behind the scenes. This learning process will go from the bottom, the basic level of Bloom's taxonomy, to the top of this pyramid. And the process goes from understanding and remembering terminology to then learning how to apply these tools to use information in new situations, then draw connections among ideas, and eventually be able to evaluate and even create new analyses using the tools that you will be introduced to. And these are just several examples that lead to more in-depth review of key topics, technologies, and analysis methods. So the program, being an introduction, will give you high-level introduction into all of these topics. Genomics, such as variant calling, transcriptomics, such as gene expression analysis, metagenomics, analysis of the microbiome or communities of bacteria and viruses, epigenomics, regulation of gene expression using non-genomic coding, and an introduction to data science. How do you apply data visualization, analysis, and machine learning to study these complex data sets and integrate them? Importantly, after this overview, you will be introduced to the concept of Omixlogic Projects. These projects are focused around the areas of oncology, infectious diseases, neuroscience, and agriculture. And you will be able to pick a project that you can follow as a tutorial or potentially design a project of your own after the program is over. You will also be introduced to the T-Bioinfo platform for big data analysis, where you can run analysis of large data without any technical challenges, understand the logic of specific pipelines, and produce different usable results that you can visually explore and understand trends in the data that you have. So to see the full schedule, we welcome you to stay in the session, ask questions, and really go through each session so that you can start your journey in bioinformatics.
Thank you so much, Ojas, for playing the video for us. Now that you know about the Introduction to Bioinformatics coursework, it's time we learn more about the next program, that is Transcriptomic Data Analysis. And to talk more about it, I would like to invite Dr. Raghavendran. Over to you, Dr. Raghav.
Thank you. Thank you very much, Sonalika. Let's get my screen sharing going. Can you see my screen? I hope, yeah, I got confirmation that you can see in the screen?
Yes, sir. We can see your screen.
Okay, perfect. Then we are in sync. Oh, sorry. Okay. So hello, everybody. One more time. I'm Raghavendran, Omixlogic mentor for Big Data Bioinformatics. And I welcome you all to this part of the webinar where I'm going to introduce you to one of the Omixlogic specialization training program, Transcriptomics data analysis. So in this program, we will learn how to use transcriptomics data. Just a second. Yeah. Transcriptomics data to understand the biology of the system from multitude of dimensions. To cite a few, the transcriptomics data analysis can identify and validate disease targets. We can explore the variation of gene expression pattern in the natural systems biology context, and etc., etc. So technically, you will learn to employ different analytical methods and reference databases to understand how gene expression vary across many different conditions. To do so effectively, you will also learn how bioinformaticians generally leverage big data processing methods that are organized into bioinformatics pipelines. You will also learn to perform exploratory data analysis, normalization techniques, and apply statistical methods for rigorous hypothesis testing. And in the end, you will also be introduced to advanced statistical methods like machine learning algorithms that is employed when the complexity of the data that you deal with increases multitude. So now a lot of this is based on omics data, which can be studied using omics technologies like next generation sequencing technologies to explore the roles, relationships, and actions of various types of molecules that make up the cells of an organism. The term omics can refer to different types of data that can include phenomics data, meaning the phenotypic data that can refer to the cell morphology, for example. We can also talk about genomics data to characterize significant variations in nucleotide and amino acid sequences. Another type of data that is growing rapidly is the epigenomics data, which deals with the non-hereditary regulation of gene activities that act without or that act independently of the changes in genomic or gene sequences. And finally, what we will be focusing on in this program is transcriptomics data analysis, which is an important part because it provides a real in-depth view of how we can use gene expression and isoform expression patterns to understand cell phenotypes. So transcriptomic studies can be performed by measuring the RNA translation of a cell in any given instant. So to measure the RNA translation, transcriptomic studies detect the mRNA expression inside the cell as an accurate alternative to measure protein expression. This allows for molecular precision in diagnostics and even development of treatment based on the molecular pathways that we are characterizing, that are understanding. So the mRNA measurements can be performed using targeted technologies like microarray technologies, which provides measurements for probes targeting a set of genes, or a more advanced high throughput technology like next generation sequencing technology that measures all the mRNA that is present inside the cell. So this technology, RNA-seq technology, has revolutionized the way in which biologists examine the transcriptomes and has been extremely successful successfully applied in biological research, in drug discovery, in clinical development, to name a few. So this can actually measure the expression levels of thousands and thousands of genes simultaneously and provide insights into functional pathways, regulatory networks in a system's biological context. So in addition to this, RNA-seq technology and RNA-seq data can provide novel insights into alternative splicing, unannotated exons, novel transcripts, to name a few. Most importantly, such a detailed analysis method and such a detailed examination will yield significant discoveries in many research areas and has already proven to be yielding more and more novel discoveries in many research areas, for example. Yeah. So we will talk about in this program. So what we will talk about in this program is our ability to leverage this kind of big data and see the significance of gene expression patterns in association with clinical data. This can help us identify molecular biomarkers for diagnostics and for therapy selections. So new discoveries include better understanding of complex diseases at subcellular level, as well as target identification for novel drug discovery and repurposing of the drugs that is already in circulation. So in addition to this, you will also find many interesting and important biomedical projects that can leverage RNA-seq data for diagnostics and cancer biology, to understand the diagnostics and biology of the cancer, and for disease classification and patient stratification, which are also important for clinical trials, and for understanding of real patient population that need to be treated, as well as using employing single cell and tissue composition in a heterogeneous tissue environment. So in this program, we will introduce you to several exemplar projects that we have prepared using publicly available RNA-seq data to study, for example, the role of tumor microenvironment in immunotherapy, to study the effectiveness of drug repurposing in mounting defense against pathogens, and to understand different types and subtypes of tumor tissues, and to employ disease stratification to accurate diagnostics, and also to perform patient stratification to understand the response of the patient to typical treatment or to different treatment options, and even to find marker cells and tissue types in neurodegenerative diseases. So the basic logic behind these types of projects and these types of analysis is to identify differentially expressed genes in an altered state or in an altered disease condition versus control experiments. And this will help us to analyze and to identify and to find these genes and to find markers of cells and tissue types that can be associated with the different phenotypes of the samples. So to help you master the transcriptomics data analysis, we will cover these topics, and these can be the topics that can be categorized into 6 major steps. We will be combining the logic of analysis with practical hands-on assignment that will allow you to understand and practice methods of analysis and types of interpretation that we can perform on RNA-seq data on real-world examples. So in conclusion, you will also be able to plan how to take what you have learned and apply to an independent research project using tools and methods we have covered in this specific program. So we will combine, for this program, we will combine online sessions, self-guided study material, and practical assignment for an immersive experience that has already proven extremely effective in our respected Omixlogic program and very well known other experiences. So the Is Bioinfo platform is designed with the vision to make bioinformatics accessible and useful without the requirement to learn coding that exclude non-experts and make conventional tasks extremely burdensome. So in addition to online courses, all participants will get access to hands-on exercises and data sets for practice. And these can be completed without any coding, as I mentioned, using the analytical T-Bioinfo platform that provides an interactive learning experience. The platform itself is color-coded to help establish a logical connection between theoretical aspects and different steps of data analysis methods that you will be explained and that you will be introduced to. These practical steps needs to be, these practical steps that are needed to be performed to achieve biologically interpretable results that we will also get our hands on. So the output of these analysis pipelines are offered as an interactive visualization or offered as a means of tables and graphics that will help users to understand the way data can be used to understand patterns and can help you start asking data-driven questions about biological phenomena or about some specific clinical condition that you are studying. So we welcome you to join this program and earn a certificate of excellence in transcriptomics data analysis. I hope to meet all of you or most of you and interact with many of you in this program. And for now, I will hand over the stage to Dr. Mohit to speak about Cheminformatics and structural biology. Over to you, Dr. Mohit, and thank you very much for your attention.
Hello, everyone. Thank you, Dr. Raghavendran. Is my screen visible? Can you confirm? And is it in full screen mode?
Yes, we can see it in the full screen mode.
All right, great. Hello, everyone. It's really good to see you all here today in this session. And today, before I tell you more about the next programs, I just wanted to go for a quick review that in April, we have started our summer training programs, which are specially designed for those who wants to start their journey into this different areas of research by using these different technologies, and the training material and the sessions that we have put together. So starting from Introduction to Bioinformatics is a beginner level course that we discussed, and Ojasvi has shared the link for the curriculum, for the entire curriculum, including the sessions, the live sessions that we have planned, as well as the resources which are associated with each of these sessions. So one of the advantages of Omixlogic programs are like the association of online sessions and the asynchronous coursework which is associated with each of this session, where all the topics that we go through and be discussed are going to also be covered in the asynchronous material, so that you can learn on your own and you can have more questions in the Q&A live sessions. So we have planned Omixlogic Introduction to Bioinformatics, Transcriptomics, and then Cheminformatics, and the research fellowship program that many of you are interested in, which is to work on projects. So starting with the first program that I am going to discuss is the Cheminformatics program. And I saw that someone here is interested in molecular docking and drug discovery and looking into the three-dimensional structures and understanding them more and connecting them with the different biological problems that to solve. So, I mean, to ensure the likelihood of the best results, researchers from the biotechnology, pharmacy, and the life science industry right now are utilizing molecular technologies that are pivotal to enable the next era of personalized medicine. So that's where we are heading. So due to this fast growth in data generation and ability of big data on public repositories, these fields of computational structural biology and cheminformatics are really evolving at a rapid pace that is enabling researchers to establish, biomolecular sequence, to structure, to function relationships using fundamental principles of physical sciences, and where these technologies are tools such as Cheminformatics, which uses algorithms and computational methods using molecular modeling and simulations to mimic biological events and dynamics. So what it is enabling is that we can now kind of model from two-dimensional sequence level to a three-dimensional model, and even think about the functional relationship and design strategies to overcome problems such as drug resistance or finding a new drug. So these ideas, I mean, how they came in for Cheminformatics, and especially in the field of graphics, has to do a lot with computer sciences. So let's not forget that this is all because of the organization, processing, and the capabilities that has evolved during the last few years. And like especially since 2020, there has been a phenomenal demand to increase the use of bioinformatics, cheminformatics, and the use of machine learning. So I think that is one of the reasons that many of you have joined this session and are interested in learning about these research topics. But just not about the understanding of the research topics, but what we have been doing at our end is to design industry related curriculums so that anyone can learn about these research topics and could use them as a skill set for their own research or for opportunities which are related to job and internships. So about the curriculum, so this program itself is designed for beginners. So anyone like students and faculty and researchers who are eager to learn about this fascinating field of computational structural biology and cheminformatics. So this collaborative training is prepared and will be delivered by leaders in the various biotechnology and pharma industry to address the need of, to address this need to introduce students and researchers to utilize public repositories and open source tools to develop quality research projects using structural biology analysis, applying cheminformatics tools. So the interested students, researchers will also have an opportunity to learn about the application of R and Python. So we'll cover that in our asynchronous session. So let me tell you more about the sessions, how they are planned. But before that, like as I was telling you, that this program will be led by mentors of Omixlogic industry expert panel, which is a very elite panel of experts from pharmaceutical industry as well as drug discovery industry. So these are the people who are really working there and have a hands-on experience, years of experience actually. So this training is designed to enable students to learn about the basic concepts of structural biology and also to start applying and working and utilizing specialized algorithms and tools that have been utilized by the industry and also the research labs to innovate, to discover and find answers to some of the important biological questions. So about the sessions, as I was telling you, that this kind of starts with a very beginner level, because one of our experiences has been that many students would want to learn this from a basic, from the very basics, and could develop really specialized skill sets, understanding the direction. So the first session, and this would be the first session, which is about the introduction to computational structural biology, where we are going to cover some of the important we will go into some of the important biological repositories. But before that, we will look into the history and techniques of the techniques involved in deciphering all these structures and involving structural biology. We'll understand protein-protein interaction, structural motifs, and the conformational folding and all of that. So, I mean, the topics are arranged in a way that you understand from a very beginner's perspective, and then grow different expertise while learning and applying Cheminformatics and structural biology. So after learning about those basic tools, like how do you visualize, how do you find data, what are the repositories like PDB, and you are looking at motifs, cath, and all of that, then we move to Cheminformatics session by Dr. Puneet Kakkar, who is an industry analyst manager at Accenture and has a PhD in drug discovery. So Dr. Kakkar will be talking about, will be introducing you to chemical interaction, chemical data basically. So it will be an overview of Cheminformatics and Cheminformatics and drug discovery pipeline, then key businesses in Cheminformatics. And then there is a hands-on session on chemical space exploration. So we have with this program hands-on session with all of the sessions where we have already prepared materials that will be shared with the participants. So in this session, we will cover some of the new technologies, discuss about what we have now with the advancements such as the introduction of tools such as AlphaFold from Google DeepMind to do the predictions more and more accurately, and also to utilize this vast database and build our own structure of need if that is required, and also to understand that how these effect of different mutations that we are learning about can be done computationally to gain insight and develop more meaningful experiments when it comes to planning experiments in the wet lab. So gaining those insights and utilizing the tools which are available, and also have a comparative understanding of how different tools work, and how some of the questions could be answered by utilizing these variety of tools with different algorithms that are meant to solve specific biological problems. So followed by that, we have the fourth session. After you learn about the structural biology basics and Cheminformatics, and then structure prediction and visualization, you will learn about this big data repositories for virtual screening basically. So there is a hands-on session on Data Warrior and visualizing for UCSF Chimera. And then there is this practical session on docking. So after learning about the repositories, before that you learn about the tools, and before that you learn about the very basics. Now you're moving to the place where we are doing experiments and creating this simulating environment to do molecular docking. So after this session, we have another session which focuses on machine learning a little bit, like how these repositories and how do you filter them properly to find and create so small repositories with more meaningful structures. I mean, many of at this stage, many of the knowledge is driven by chemistry, So medicinal chemistry, perhaps. So this information and how do AI is now playing a role in here. So we are going to use some specific tools such as Data Warrior, and you can Google it and check that out. It is a visualization tool, and also you can do a lot of filtration to find out. There are several more tools and features that are involved in it. So followed by that, like once you have learned about all of this, now it also makes sense to think about in the aspect of how does this all make sense in aspect of a project. So designing a Cheminformatics research project. So how does that work? What you need to know, and what are the different components, stages, and then challenges. And then we have another follow-up program in July and in June and July that would be more of an advanced Cheminformatics, which is to work on research projects and develop using some advanced tools and some data sets which are bigger in size. So all of these programs that we have discussed has a support through the asynchronous coursework. So that will be available with this, and also the expert sessions. So there's an expert session on drug repurposing by Dr. Uday Saxena. He is a co-founder of a startup company called Regen Innovation, and he has huge experience of working in big pharma companies such as Pfizer, and worked with a team that discovered Lipoter. And he has been working with us for a while in helping students learn about different aspects of drug repurposing and drug designing with all of his experience. So he'll be talking on 21st April. It's a tentative date right now, but yes, I mean, we have this session coming up on drug repurposing. Followed by that, we have another session by Dr. Ajit Kamath, who has 25 years of experience on preclinical drug discovery. And he'll be talking about the advancements that he has been following with the drug discovery, thinking of the huge experience that he has, and also looking at what is going on and how this entire industry has evolved from doing virtual screening and high cost screening to now more targeted screening and with less and less cost. So he'll be also discussing about precision medicine, which is one of the key and hot topics in healthcare right now. So after this session, we have an industry panel group where we are going to discuss about the job opportunities. So many of these innovators and leaders are also recruiters, and they will be discussing about the expectations, and we will have an open stage, and we will discuss about the skill sets that one would be requiring to look for such opportunities. So in April, as again, I would say that we have these programs planned for those who are interested, but these will be also available throughout the summer. And anyone who is interested and is not available right now could also join these at a later stage in the summer, where we have all other plans and other programs which will be also piloted and would be available for anyone to go through. So one of the program which is which helps you perhaps to grow from training to develop and working on a project is research fellowship. But for beginners, we have a lot of content on the T-Bioinfo platform and also on the Learn OmicsLogic that we have been discussing. And actually, these are a great start for anyone who's a beginner, and also for students and also for faculty to brush up and utilize many of these lessons into their teaching and into their curriculum. So similarly, we have a curriculum from one of the faculty which has been now uploaded on the platform, and I would appreciate if someone can share the link of these courses that are free to take and to gain basic understanding, and then go for the different mentor-guided training. So this is how the entire pathway looks. From basic training that I just discussed, we have advanced trainings such as the ones that were discussed before, and then we are discussing about specialization, project work that Ilya described in such a beautiful manner. And then we have the projects and internship. This is the summer research fellowship program that we are really excited about, because this is not for a beginner, but also we get many students who want to do work on a project and are beginners. So we have different modules that are from starting from 45 days to 6 months and 1 year, where you're working on different such big problems, challenges, and coming up with a research project, publication, and all of that. So we have been working with a lot of students and fascinating projects. Many of them we were able to complete, and many of them are online. So I'll just quickly tell you that how it is, what it is all about, without not taking much of your time. So we are in these programs, or in this program, where it is a research internship or a research fellowship, where we are working with students who are utilizing the T-Bioinfo server and learn Omixlogic for doing and developing a project. So it all starts from processing, analyzing, annotation, integration, model, and simulate, where we are looking for samples and data from online repositories. That could be patient data, that could be animal model data, that could be a cell line and tissue level data. And then there are availability of phenotypic data, next generation sequencing, and all of this. So using all of this data, we are trying to find answers. And in this entire workflow or entire pipeline, we are developing skills, experience, connection, and validation, most importantly. So the work that we do usually does have a lot of similarities with drug discovery and pharma industry, and also healthcare. So drug discovery and clinical data is something that a big data approach would consider. For example, precision medicine is one of such example. So our focus is to help anyone who are interested in such proposals, who are interested to work on research projects based on their own research ideas. And our team actually helps them develop that project into a real set of goals, objectives, and then the analysis, interpretation, and then writing. So this entire process is for students, it's really beneficial, because what they get from this is real experience working on real data sets, facing the challenges, writing and developing such skill sets to present and also to record their experience, and for also getting an opportunity to work on industry projects. So many of our research fellows are also hired for industry projects, where we are working with several of these research labs and industry, where we are working on big data sets. And there are a lot of opportunities on those projects that students get after learning about this entirely and doing it themselves by one first-hand experience. So the research consultation is a weekly research consultation that we do, where we have cohorts for different research based on different research interest, based on university batches, based on specializations such as neuroscience. So we have an Alzheimer's group, where a group of researchers and students are working on exploring data sets and finding answers. We have a space omics research group. Similarly, these are also like independent projects that anyone independently can work on. So we have a lot of independent participants who are working on their research projects and doing some work on a specific for a specific degree or for a specific training. For example, with MIT Noida, we have this NTCC program and also other research internships, where students are completing their research internship as well as getting recognition and collaboration with their faculties, leading to more outcomes such as project publications and henceforth. So we have a lot of participants who have completed without any knowledge of, they started their journey without any knowledge of big data, bioinformatics, and all of that, and they were able to really do really well within a short period of time. So even we have students who completed their research in 3 months period of time, and students who were able to complete their project in 6 months period of time, but after completing their basic training. So the basic training is very important for this to get some of these achievements, to get some of these outcomes. So I would recommend you to follow the path that we demonstrated, and that could help us to help you more and more. And if you are already working on one of these areas and you have a prior experience of bioinformatics, then you can directly work on a research project. So for beginners, there are resources and training available. For intermediate, there are resources and training available, and for advanced, also there are resources and training available. So we similarly have plans in May for where we are going to pilot these programs, getting started bioinformatics in R, Python, space omics, infectious diseases. So I'll be looking forward to meeting you again. If you have an opportunity to join any of these programs, that would be great, because these are the basic programs and where you start developing all of these skill sets and explore more. And throughout the summer, we will be working on research fellowship. So research fellowship is one such program that you can join anytime. There is no time limitation, but the limitation there is the skills. Like if you are at an intermediate level, that would be the best time to join the research fellowship. If you have gone through some of the basic material and understand how to utilize the T-Bioinfo server, gone through some project examples yourself, because we have prepared them with one objective. That objective is to make you to prepare you for all the challenges that you are going to face in this research journey. So I hope this information was useful for many of you, and we will see you together collaborating on some research ideas and training. So it was really great to see you all. Now I would like to pass this stage to Sonalika to help you with the registration, or at least to show you how the registration process works.
Thank you so much, sir. And now I would like to take you all one by one. Let me share the link. Yes, I'll be taking you all on the program page of each one of the program that we discussed today. So this is the first program page, that is Introduction to Bioinformatics. Now, those who are interested to get themselves enrolled for Introduction to Bioinformatics program to get started with bioinformatics and the topics that we discussed today. So this is the program page that you can visit and learn more about the program schedule. Now, when you scroll down, you'll be able to look at the schedule of the whole program that we have designed for you to get started with bioinformatics. So we have 6 sessions in total. The dates have been mentioned, the session title has been mentioned, the topics, and also the associated resources are mentioned. Now, what are these resources all about? These resources are actually lessons of various courseworks on the portal itself. And just after explaining, after taking you through the program page, I'll be also taking you through the portal and tell you how you can navigate the portal and make the most out of the resources that are available there. Okay, so we have the first session on April 11th, and then the second session is on April 18th, which is about Introduction to Genomics, Mapping, and Sequence Visualization. And the third session, in the third session, we're talking more about transcriptomic data analysis, that is on April 25th. On 2nd May, we'll be taking you from an understanding of transcriptomic data analysis to differential gene expression analysis and also the biological interpretation of the data. And then on May 6th, we'll be talking more about Introduction to Biomedical Data Science and learn some aspects of machine learning in application to various types of data that we would have been discussed till then. And also then we'll talk about bioinformatics project examples based on machine learning for NGS data on May 9th. So we'll be touching upon all the various aspects of multi-omics data types, and also we'll be discussing about the techniques that could be applied to analyze and work with these different omics data types and interpret some meaningful biological insights out of this data. Okay, now how do you register yourself? So to register yourself for the Introduction to Bioinformatics program, if you scroll down, you'll be able to look at 3 different levels, or I'll call it subscription levels. We have one level that is a beginner level. Now, this level will give you the access to the live group sessions that we'll be having. And also you will be able to take your questions, be a part of a community and get all the resources, access to all the resources. An intermediate level will give you an access to one-on-one mentor support. rather than group meetings. You'll be able to schedule one-on-one live meetings with your mentor and take your questions ahead. And at an advanced level, you'll get some extra time to work on your research project and also have a research proposal in hand that could be taken ahead and give you some really good outcome that could either be a research publication, a poster presentation, or a project to be submitted in your college if you are a student from a bachelor's or a master's degree as a requirement of your last semester. Some of them have it as a requirement to complete their last semester. So you will get some extra time to learn a program and then apply it to a project, maybe to sit with the mentor and have your research project designed. Okay, so these are the 3 different levels, or I'll call it subscriptions: beginner, intermediate, and advanced. Now I've explained these 3 subscription levels, so I'll keep it constant for the other programs as well. I'll just take you through the syllabus of the other 2 programs that we discussed about. So then we have Transcriptomics program, and then we have the Introduction to Cheminformatics and Drug Discovery program as well. Now, in the Transcriptomics data analysis program, the program starts on 12th April and will end on 12th May. And the schedule of the program is mentioned on the program page. Now, again, there are 6 sessions for the program as well. You can go through the schedule of the program here. And again, we have 3 levels: beginner, intermediate, and advanced, where you will get some extra time to work on your, propose a research question in the advanced level, get one-on-one mentor support in the intermediate level, and in the beginner level, you have the access to usual sessions. And then coming to Introduction to Cheminformatics and Drug Discovery program, this program will be starting on 19th April and will end on 19th May. And the schedule of the program is again here on the program page. Let me just share the link in the chat box. And you can learn more about the schedule on the page itself. And talking about the program enrollment, again, the same 3 levels are here too. And to pick the level that you would like to opt for, you can click on the select option button and enroll yourself. Now, I would like to quickly take you all to the portal login button first of all. So those who have not yet created an account for themselves on the portal, they can do so by clicking on the login button. It's very easy to create an account for yourself. Let me just log out and give you a quick demo. You need to just click on the login button. Now, either you can create an account for yourself simply by clicking on create an account and putting in the details that are asked, or you can also create an account for yourself or sign up by opting any one of your social media accounts. I'll be doing it by clicking on my Google accounts, and this will help me log in. I'll also put the link to the portal in the chat box. Thank you so much, Ojasvi, for the link. Okay, now once you have signed up on the Omixlogic Learn portal, the first thing that you need to do is update your profile. So click on the email ID in the menu bar and add a profile picture. Your name should be in title case. Make sure that your name is in title case because this is the same format that will reflect on the certificate. Please connect your social media accounts and add a brief bio about yourself. A brief bio could include your educational or your research background if you are a researcher, and also your research interests that you would like to work on. And once you are a part of any of these 3 programs that I just showed you, the program pages, you will be able to see the badge of the very same program on your profile. Now, clicking on this badge will take you through the program page. Now, also on the very same program page, you will be able to look at the recording of the session here in the page itself after the session is completed. Now, let's say you're not able to attend the session live because any of your commitments, or you want to revisit the topics that were discussed by the mentor in the session, you can get back to the program page and look at the recording. The recording will be there for you throughout the program. Okay, now coming back. Now, all the lessons that I showed you, I was talking about the associated online resources, and I told you that these are actually the lessons on the Omixlogic Learn portal. Now, where are these lessons located? And how can you monitor your progress with these lessons? So this is the place, the profile itself, where you can see your progress, what you have been completing or working so far, and also you can see how many lessons you have completed for each of the courses and get a certificate once the full course is completed. Now let's look where these courses are actually located. So if you click on the courses button, you get a list of courses, example projects, which are based on various omics data types, which includes transcriptomics, metagenomics, epigenomics, and genomics. And then you also have various example projects. Now, these example projects are actually designed to give you an essence of how the type of omics data you learned about could be applied to answer, the techniques that you learned about could be applied to answer a research problem in hand. So now how do you go through the various courses that are present on the portal? Just click on the courses tag, which is here on the right side in the various tags, and clicking on the courses tag will help you look at various courses that are there on the portal, right? And you can get started with the course of your choice. We recommend that you get started with Introduction to Bioinformatics for those who come from a non-bioinformatics background. And then also we have a coursework that is Bytes and Molecules that you can go through and get introduced to various omics data types and specialization tracks. And last but not the least, as I was talking about the example project, so if you just click on the project, you will be able to have a look at all of these example projects, which are actually the applications of various courseworks here on the portal. So that was it about how you can navigate on the portal. And if on any of the links that we share in the chat box, but you would like to revisit it later and enroll for the program, then you can click on the programs icon in the menu bar itself, and you'll be able to look at the various programs here and simply click on it. This will redirect you to the program page and get yourself enrolled to get started. So that was it. If we have any questions, you can please put them in the chat box and we are here to guide you through. Okay, so Himanshi is asking about the fee structure for the research fellowship program. Okay, so for the research fellowship program, Himanshi, you need to visit the research fellowship page. You just need to click on the research button here, and this will take you to the research fellowship page. Now, when you click on learn more, you will be able to learn more about the duration of the research fellowship program that you can enroll for, and also the structure of the program. And if you click on get started, you will have here two subscription levels, beginner and advanced. Now, in the beginner level, you will be able to get the access to the education cloud pipelines. And in the advanced level, you will be able to get the access to research cloud pipelines. And there's a slight difference between these two subscription levels. So in the beginner type of subscription level, we generally refer to the various big data repositories that are present. So we extract data from these repositories and we work on them, analyze and interpret the results, right? And in the advanced level of subscription, that is more recommended for people who are actually generating their own data, maybe if they are a PhD fellow or postdoc fellows and they have their own data which is generated in their labs and they would like to work with it. So what happens in the advanced level, we give them an FTP account where they upload their data and then they work with it in their research project duration, right? So you can figure out whatever requirement you have based on the type of data analysis that you would like to take further for your research project. And that is the slight difference that I told you. Okay, now there are two options. Either you enroll for a 3 months program or you enroll for a 6 months program. Now you can see the 3 months subscription levels like here. And if you pick a 6 months one, then you have the options placed here. Now, the thing is, if you want to get enrolled, first of all, you can mail at marketing@omixlogic.com. Either you can enroll yourself directly, or if you opt for a scholarship, then you can fill out the scholarship form. I'll just take a second and I'll share the scholarship form in the chat box. Yes. Here is a scholarship form. You can fill out the scholarship form and our team will review your application and then we'll get back to you on it, right? You can also mail us directly at marketing@omixlogic.com. Is that clear, Himanshi? Okay, thank you. Do we have any more questions? Yes, we have a question. I think the person has not named himself or herself and has the name by the device. Yes, you can definitely go through all these resources without a Linux system, not without Windows, definitely. You need a browser to go through these various resources on the portal. Yes, Nadia, the sessions are recorded. If you have an internet connection instability issue, you need not worry about it because we have everything recorded and we'll share the recording with you. And also we have Slack channels. So you can go through the recording and you will be a part of a Slack channel and you can take your questions there in the group at your own pace. You can go to the recording and whenever you get time, you can just put your question about the session in the group and the mentor will reply to you based on their availability. And we can also take your questions in these live sessions, definitely. Reshmi, to get started, you can go to the courses tab and there are various courses here on the portal. Let me just show you the free resources to get you started because we have a lot of students and researchers as well who come from different fields and not computational biology specifically, but they wish to get started with computational biology at some point in their research or to begin with their career. This is one particular course, that is Introduction to Bioinformatics. This is a free of cost course which you can go through and get a sense of what is bioinformatics and what is its role in translational healthcare and why should you actually bother doing bioinformatics, as simple as that, right? And the other coursework is Bytes and Molecules. That is again absolutely free of cost. In this coursework, you will be able to understand these various omics data types that we discussed: genomics, metagenomics, epigenomics, transcriptomics, and also some of the specialization tracks like precision medicine, space omics, and also infectious diseases, and get started with R. And also, there are various other courseworks too, in which few of the lessons, like first few lessons are free of course that you can go through. And except that you have some start lessons for which you would be required to be a part of the program and take mentor guidance to understand these technical concepts. Realme Pad, yes, you can learn from your device, definitely. The way Raghav sir and Ojasvi have explained it to you. Hiba Kasir, yes, you need to enroll yourself for individual summer program, whichever program you are interested in, or the research fellowship program. And because taking the subscription level will help you get the access to the resources that we have discussed so far, and also to the mentor guided sessions. Okay, so do we have more questions? If we have any questions, please put them in the chat box so that we can answer it for you. And if not at the moment, then you can anytime feel free to please mail us at marketing@omixlogic.com.
Can I chime in for a while, Sonalika? I think
Yes, sir,
there's a little bit of confusion over what I'm posted in the chat. So the lessons can be learned. You can go over the lessons. You can understand what the lessons tell you, what the asynchronous materials tell you. Even you can go over the recordings also. Also all the resources using any device that you can get your hand on. But to completely understand the logic behind these analysis, you will have to set up pipelines. You'll have to understand. You'll have to upload the data, or you'll at least have to upload links to the pipeline and preferably download the output files. Open these tables, output files, which could be tables or which could be pictures on your device, and then perform some sort of basic analysis like filtering, like sorting. So these filtering, sorting, and to explore the tabular output that the pipelines are giving you, or are giving access to you, cannot be performed, unfortunately, on every tablet that you can get hands on. On some really high-end ones, you can actually download the tables and then open it up and do some basic analysis like filtering, sorting, and some very basic plotting. That's what we do. But for these, yeah, I think it is recommended that you get access to computer, at least at the phase where you are running pipelines and trying to copy or trying to download the output files and exploring the output files as you follow along with the synchronous or asynchronous materials. So that's what I wanted to clarify, because there has been multiple different versions of answers that we actually told you. Thank you very much. Over to you, Sonalika.
Thank you so much, sir, for the clarification. And I have a question in the chat box regarding the certificate. Yes, all these programs are certified. So there are two possibilities for the certification. Let me share my screen and go back to my profile as I was talking before about the certification process. So you see, when you complete a particular program, once the program is completed, so I'm just giving you an example of the previous programs that we had, like workshops, symposium, etc. So once these programs are completed, you can view your certificate in the in your profile itself. Just click on the view certificate button. I think because of minute is taking time, and then simply download it. Yeah, it's up. So simply download this, and then you can share your achievement wherever you would like to. So that was about the certification process. And do we have any other questions? I think some participant has, please keep yourself on mute. If you have any questions, then you can unmute yourself. Okay, so if we have no more questions.
I think I see one more question which asks about online softwares aren't enough to analyze for the analysis. So more often than not, what you see in an online software environment is the softwares. I mean, let's not call them softwares. Most of the methods that you find online are actually tailored for that specific data set or that specific source organism, and they're not generalized as such. So what we actually offer you is our analysis methods or our way of categorizing these multiple different pipelines, multiple different algorithms and softwares, and then giving them to you so that you don't have to worry about n number of different settings that you have to set every time that you want to install. So almost all of the algorithms that you see online for omics data analysis involves installation and understanding of at least 4 or 5 different algorithms with as many as different parameters. So what we offer you is a set of different pipelines that will take specific paths or specific sequence of these algorithms that can be employed for the different omics data that you can analyze with. So what the bioinformatics big data bioinformatics platform gives you is, it will give you options to set parameters only that are necessary to be set for the omics data. So you don't have to worry about the other parameters at all. And if at all, if you want to set these parameters, then we give you explanation on what is the expected number or what's expected value, and where are you expected or where are you required to find this specific parameter and set these parameters. So these information that you will not find easily on the online softwares, and more often than not, you will have to try 4 or 5 different sequences, and in the end you are going to spend a lot of time trying to read the manual and trying to curate different algorithms for the data that you have. So that's basically main difference between what's already available out there and what we actually offer you. So of course, as I mentioned, you can take the Bowtie algorithm and then try it out on your computer. But of course, to run Bowtie, you need at least a computer with 64 GB or 128 GB RAM. So you don't have, most likely we might not get access to those high performance computing. And even if we have very competent computers, then you will have something like, you have to install the softwares, manage dependencies, and trust me, I've gone through all those different routes, and it's not fun trying to solve dependency issues. So yeah, and I can hand it over to Sonalika again if you have any other queries. So much. And somebody asks if there is a certificate issued.
Yes, I have taken that question.
Perfect. Thank
yes,
you.
Sir. Okay. So thank you so much, everyone, for joining today's webinar. And for any queries, please feel free to reach out to us at marketing@omixlogic.com. And we also share with you the recording of today's webinar. And it was nice to have you all with us. And you can get back to us for getting enrolled and getting started. Thank you. Bye-bye. Have a nice